Good afternoon. Hope you enjoyed lunch. Welcome to New Insights in Lipid Metabolism and Atherosclerosis. I'm Peter Toth with my principal academic affiliation being with Johns Hopkins. And I'm joined here with Sharif Abdelkarim, who joins us from Casablanca. And our first talk is going to be by Professor Alan Tall, who's going to talk to us about new pathways linking cholesterol efflux to inflammasome activation. And I'm sure he's going to show us a great deal of extraordinary new science.
Sharif. Thank you so much, Jean-Charles and Jamila, to invite me for the second time, actually, to beautiful Marrakesh. And yesterday was a beautiful day, and my wife and I were out looking at the sites, and it is just such a delightful city with so much interesting culture and history and background. So thank you. We worked for many years in the area of lipoproteins, particularly in HDL. And in recent years, we've been attempting to link studies of cholesterol and lipoproteins to inflammatory processes, given their important role as summarized so beautifully by Peter earlier today. And so I'm going to touch on mechanisms of cholesterol efflux and how they may be linked to inflammasome activation. Just to start off with a piece of clinical trial data, you will all remember that the REVEAL study with the potent CETP inhibitor anacetrapib went to its termination. It was the largest and longest running study of a CETP inhibitor, the most definitive one to date, and did show a benefit with a an odds ratio of about 0.91, moderate perhaps, but with a very good p-value, so a great deal of certainty around this result. Many people feel that since the degree of benefit appeared to be roughly what would be expected based on the degree of LDL cholesterol lowering, which was about 20% based on centrifugally isolated LDL, that the benefit could be explained largely by the lowering of LDL. I don't think we can really clearly make this conclusion from a clinical trial, but certainly the study would be consistent with the benefit of LDL lowering and didn't uncover a major adverse effect of raising the HDL, which some people had suspected might happen. As so nicely described by Raul, the movie is not yet over. It's going to replay with another CETP inhibitor called obicetrapib, which does appear to be more potent and, most importantly, lowers LDL cholesterol by about 40%, centrifugally isolated LDL, not a calculated number. And that corresponds to the LDL lowering we first described in homozygous deficiency of cholesterol ester transfer protein, suggesting that it is effectively, completely inhibiting CETP, and this will lead to a clinical trial, and so hopefully the curtain will not close on this area. So what is the HDL particles, the result from CETP inhibition, we found to be effective at mediating cholesterol efflux, but the studies that were actually done with statin plus anacetrapib show a fairly minimal increase by the radar group, Dan radar group. Nonetheless, one of the ideas, principal ideas supporting an anti-atherogenic effect of HDL derives from studies of cholesterol efflux where plasma or HDL particles are taken from a patient's blood and put on isolated macrophages, and then the rate of cholesterol efflux is measured. And that correlates with both cross-sectionally and prospectively in most of the studies with a benefit. The higher the efflux capacity of HDL or plasma, the greater the benefit. And those studies were really done mostly by the Radar Lab. We did a study that used our own cholesterol mass efflux assay, which may be more specific for the physiologically important net efflux component than the radio-labeled assay. Whether or not that's true, we did see a benefit on coronary artery disease in the prospective MESA study. So these were baseline samples, and we're looking at incident coronary artery disease. But interestingly, did not show any benefit in this for non-hemorrhagic stroke, so ischemic stroke. And whether this is a biological difference or whether it merely reflects the complexity of a clinical stroke classification is still not clear. The cholesterol efflux from macrophages is principally mediated by two ABC transporters, ABCA1, which you all know is the mutant molecule in Tangier disease and results in foam cell formation, macrophage cholesterol accumulation through the body, and as we discovered, a second transporter, ABCG1, that seems to promote efflux onto HDL particles, and so lipidated ApoA1 by ABCA1 can develop into a mature HDL particle. And we, through the years, have examined the impact of deleting these transporters in a cell-specific fashion on the process of atherosclerosis, and by and large, our studies show a benefit or a harmful effect of deleting the transporters, or a benefit of the transporter activity. And we've shown that by deleting it in macrophages, endothelial cells, and hematopoietic stem cells. So in hematopoietic stem cells, the cholesterol efflux transporters actually serve to limit the proliferation and expansion of hematopoietic stem cells. And that has segued into, later on, our studies on clonal hematopoiesis. So very recently, within the last year or so, a group in Dallas, led by Zhanchan Li, and we're not involved in these studies, but they're so beautiful, I thought I'd like to show them to you, has used cryo-electron microscopy to solve the three-dimensional structure of ABCA1 in two states with ATP bound or not bound. This is the ATP bound form. This is the non-ATP bound form. And the ABCA1 structure looks like other ABC transporters in general, except that these are the transmembrane domains here. And all of this here is an extracellular domain. So ABCA1 has a very large extracellular domain, likely involved in the binding of ApoA1 or small HDL particles. Their structure revealed a cholesterol molecule within the hydrophobic tunnel in the extracellular domain. When ATP was bound, the cholesterol molecule was still in the same place. Of course, these are static structures. But that caused closure of the cytoplasmic regions because the ATP molecules bind to what's called the ABC or Walker motif that brings the intracytoplasmic domains closer together, and also the transmembrane domains also come closer together. And so Zhanchan was kind enough to share this. Next slide. We'll see. So this is the ABCA1 molecule, and this is a movie. Okay. So focusing first on the ATP binding site. So the ATP molecules bind. They cause closure here, and the transmembrane domains move closer together as well. And then when ATP is hydrolyzed, the whole structure loosens, and you can see this pumping action where likely cholesterol molecule gets pumped across the transmembrane space to the extracellular domain, and then an ApoA1 somehow likely binds here and picks up the cholesterol molecule. So my talk is about inflammasomes. And Marit Westerterp, in my group at that time, discovered that mice with myeloid deficiency, that is neutrophil and macrophage deficiency of the transporters, had prominent inflammasome activation in their macrophages. So this is a Western blot showing splenic macrophages and neutrophils together, neutrophils here and monocyte macrophages here. And what you see here is the cleaved form of caspase 1. the enzyme that Peter referred to that actually activates IL-1 beta and IL-18. And so you see that it's all in the cleave form. So it's highly activated in the macrophages with cholesterol transporter deficiency. And Merritt carried out atherosclerosis studies in these mice and found that when the essential molecules in inflammasome activation, NLRP3 or caspase 1 and 11, were knocked out, there was a reduction in atherosclerosis in the myeloid ABC1 double knockouts. You'll notice that there actually was no impact in our studies in the standard LDL receptor knockout mouse model, and that's been the finding for several different laboratories, in contrast to the findings from several other different laboratories that do find an impact in the standard model. But my thesis is that prominent inflammasome activation in atherosclerosis requires a second hit. So the first hit being hyperlipidemia, the second hit being defective cholesterol efflux, a clonal hematopoiesis mutation, or some other factor, maybe diabetes. This is a nice inbuilt system. It stops you talking too quickly. So human relevant. So these are Tangier disease patients with homozygous deficiency with Tangier disease and heterozygous. And they showed elevated levels of IL-1 beta and IL-18 in the bloodstream, which may be serum biomarkers of perhaps some value in indicating some level of inflammasome activation going on in these patients. This may actually be relevant to the peripheral neuropathy that these patients develop because there was a study in Science that used our mice and showed that when you did knockout of ABC1 and ABCD1, there was impaired peripheral nerve regeneration. Merritt also found an increase in nets, neutrophil-derived nets within the atherosclerotic plaque. So this was shown by co-staining for myeloperoxidase and citrullonated histone, giving a yellow color. So this is in the myeloid ABC double knockout mice. And then here, when you knock out the inflammasome mediators, caspase 1 and 11, you see the yellow area is much diminished. And then a series of studies conducted over, since Merritt left the lab, the last 4 years, and published recently in Cardiovascular Research, using macrophage or neutrophil-specific knockouts of ABC1 and ABCD1, we showed that macrophage deficiency of the transporters, not neutrophil deficiency of transporters, activated the inflammasome, secreted IL-1 beta, and we collaborated with Peter Libby on this. IL-1 beta antibodies were kindly provided by Novartis, and we showed that the crosstalk from the macrophages to the neutrophils was mediated by IL-1 beta, and when we gave the antibodies, we inhibited the neutrophil netosis. So the IL-1 beta actually, as Peter, and I wasn't realizing this, Peter, you originally showed that IL-1 beta is autocatalytic. It is secreted by a cell and goes back and acts on its own receptor. So IL-1 beta made by the macrophage acts on the IL-1R1, the receptor in the neutrophil, and activates or primes the neutrophil inflammasome that then releases nets. But it isn't actually cholesterol accumulation within the neutrophil. So even though neutrophils have the ABC transporters, for some reason they don't activate the inflammasome. I don't know why, but that's what we found. And then the principal signals that bring in the neutrophils are endothelial. IL-1 beta activates the endothelium, of course, and then the second effect within the vasculature is actually this activation of the neutrophil inflammasome. So that leads to perhaps a more fundamental question, and that is, what is the mechanism of NLRP3 inflammasome activation in atherosclerosis? In general, NLRP3 inflammasome activation is something that requires two signals. The first signal occurs from PAMPs and DAMPs that are present within the atherosclerotic plaque, things like HMG proteins, oxidized LDL, that interact with toll-like receptors and act via the NF-kappa B pathway to induce the components of the inflammasome. So NLRP3, procaspase 1, and IL-1 beta are all highly induced by priming. But then nothing happens until a second signal comes in. And so in a cell culture experiment, that can be done by adding ATP, and probably necrotic cells in the plaque are releasing ATP, and so that's an important factor. And then certainly in gout, urate crystals can activate, provide a second signal, and probably they can in atherosclerosis as well. And so Ika Lutz originally, and then subsequently from Catherine Moore's lab, suggested that lysosomal damage by cholesterol crystals or plasma membrane damage that are present in the plaques or formed after modified LDL is taken up by CD36 and then nucleates cholesterol crystal formation within the ER may be activating the inflammasome. And I think this likely is true. I would not say that it has the status of an urban myth, but it has an easy appeal to it that may not be the complete truth. And so why do I say that? When we looked in our model, oh, there it is. We found that we had cholesterol accumulation in lysosomes in the myeloid knockout of ABCA1 and ABCG1, like shown here by co-staining of lysotracker and philippin to mark cholesterol in the lysosome, the yellow color, but no cholesterol crystals. So we're getting inflammasome activation without cholesterol crystals. And so we did another experiment to more precisely look at this question. And so we fed modified LDL to a macrophage, and we also treated with an inhibitor of the Niemann-Pick C protein 1, which is the one that promotes the efflux of cholesterol from lysosomes into the remainder of the cell, the inside of the cell. And we found that when we treated acetyl LDL and gave this U compound, so-called, we got refractile material in lysosomes, these little white dots here, actually microcrystalline cholesterol, but that was not associated with inflammasome activation. So when we treated these cells with LPS and ATP, there was no inflammasome activation. So this experiment dissociated cholesterol crystal formation from inflammasome activation. So I'm not saying that cholesterol crystals might not activate the inflammation. They probably do in advanced atherosclerotic plaques. But what I'm trying to say is that there's another mechanism going on, something else that cholesterol has to get inside of the cell to do something else. So what is that something else? That's another mechanism potentially was suggested to us by basic work done on the NLRP3 inflammasome. And this group here had done a chemical inhibitor screen and come up with an inhibitor that blocked inflammasome activation and discovered that it was working by inhibiting what's called a priming event, which is a TLR JNK1 phosphorylation event on NLRP3 that is required, a specific serine residue that's conserved in humans and mice, has to be phosphorylated, that then leads to a deubiquitylation event and allows assembly of the NLRP3 inflammasome. So we chased after this and discovered, and this is a complicated slide, we discovered that when cells are loaded with modified LDL or have knockout of the cholesterol efflux transporters, cholesterol accumulates, yes, in lysosomes, but then goes to the endoplasmic reticulum, just like many other cholesterol homeostatic events, and then activates by the IP3 receptor, calcium release, CAM kinase 2, and JNK, a cytosolic complex which has been shown to be the one that phosphorylates and deubiquitinates NLRP3 by the earlier work that I showed you. So this pathway gets activated when cells are accumulating cholesterol. And so what's the proof that it's important? So I'm not going to show you all the individual experiments. These experiments have been led by Mustafa Yeltsin-Kaynar, who's a brilliant Turkish postdoctoral fellow in the lab. So there's a specific inhibitor of the BRCC3 deubiquitinase, the enzyme that deubiquitinates NLRP3 to allow it to become activated. And we carried out an atherosclerosis study with this drug called holomycin and found a reduction in lesion area, a trend to reduce necrotic core area, And then an increase in fibrous cap thickening. You all know that mice don't rupture their atherosclerotic plaques, but I was delighted to learn this morning about two imaging studies using OCT from Raul that have shown that a PCSK9 inhibitor is actually thickened by doubling the thickness of the fibrous cap, correlating with protection from MACE. And so even though mice don't rupture their plaques, I do think this could be a sort of valid endpoint of plaque thickening, of fibrous cap thickening. And in addition, and this follows since I showed you that netosis is downstream of inflammasome activation. So netosis was much improved by this drug, holomycin. In the last few minutes, I will turn to a discussion of clonal hematopoiesis and then try to link it to the first part of the talk. And so clonal hematopoiesis was discovered by our colleagues in the hematology community, particularly Ben Ebert, Sid Jaiswell, and Alex Pick, and some others. And they discovered that in some situations, mutations would arise within hematopoietic stem cells. Please go backwards. It's just going forward. Okay. And these mutations endow a proliferative advantage to hematopoietic stem cells that then results in clones of white cells in the blood containing the mutation. If additional mutations occur, this can give rise to leukemias and, of course, very serious. But much more commonly, single or double mutations occur in the blood in the setting of what's called clonal hematopoiesis. And Ben and Sid discovered that this increases the risk of atherosclerotic cardiovascular disease. Some mutations increase venous thrombotic risk and also increase incident and progression of heart failure. This is from Sid Jaiswell's New England Journal paper where they studied several populations and identified increased coronary artery disease risk in association with mutations in several different hematopoietic genes, DNMT3A, TET2, ASXL1, JAK2, and some others here. And mostly these are epigenetic modifiers. JAK2 mediates signaling in response to hematopoietic cytokines, and particularly increases atherosclerotic risk and venous thrombotic risk. So we've studied particularly JAK2 clonal hematopoiesis and also TET2 clonal hematopoiesis, both of which have been linked to inflammasome activation. Trevor Fiedler in my lab, a postdoc at the time, discovered that the JAK2 mutation actually predisposes to activation of the AIM2 inflammasome, different to the NLRP3 inflammasome I've been talking about to this point. AIM2 is activated by fragments of double-stranded DNA that are recognized in the cytosol by the AIM2 mechanism and lead to the whole program that's similar for all the inflammasomes, leading to gasdermin D cleavage, pore formation, release of IL-1 beta and IL-18, pyroptotic cell death and cap thinning. So he showed all of this in a mouse model that atherosclerosis was increased with features of plaque instability. And very recently, and this is published as a preprint, Ziyu and Pradeep Natarayan at the MGH Broad have looked at the question of what inflammatory genes modify the risk of CHIP mutations. And what they discovered was that the genetic factors that are predicted to increase the expression of the AIM2 inflammasome greatly enhance the risk of JAK2 CHIP for coronary artery disease, providing a strong confirmation of the predictions from our mouse model that was published in Nature. But in addition to that, they discovered that ASXL1 CHIP, ASXL1 it's an epigenetic modifier. Also, risk was predicted by the AIM2 level of expression of the AIM2 inflammasome, and Trevor in the lab used CRISPR to make a ASXL1 chip mouse and confirmed these observations in the macrophages from those mice. And then overall, Peter was also involved in this study and noticed that in addition to that, all of CHIP mutations were more likely to increase CAD risk in people who had increased expression of the IL-1 receptor-associated protein that actually increases IL-1 signaling in target cells that have the IL-1 receptor. So this sort of really indicates that a second hit with a CHIP mutation may be an important factor in increasing cardiovascular risk mediated by inflammasomes and IL-1 beta. And so I think the field is developing, I hope develops into an area where precision medicine targeted approaches to anti-inflammatory therapies may lead to a better benefit to risk ratio by targeting people who are at greater risk and therefore may have a better therapeutic index. An early study, one of the first study, actually the first study that was done of a CHIP mutation in a mouse model, and then also Peter and his colleagues did a very similar study at about the same time, was carried out by Jose Fuster and Ken Walsh, and they showed that a TET2 CHIP model had accelerated atherosclerosis, quantified over here, and that an inhibitor of NLRP3, MCC950, actually reversed that effect. They noticed no effect in the basal model. You need the second hit of the CHIP mutation, then you see the effect. And we have also been studying TET2 clonal hematopoiesis and found that the activation of the NLRP3 inflammasome, as assessed in splenic cells and splenic macrophages, that it requires both hyperlipidemia, hypercholesterolemia, and a TET2 mutation. So you need these two hits in order to get the inflammasome activation. You need cholesterol, and you need a TET2 mutation. So here is cleaved caspase 1 quantified, and it's only really significant when you have both hits. And we could reverse that by infusing reconstituted HDL. So this is the forerunner of CSL112 that several of you are probably involved in clinical studies that are ongoing at this time to see if it reduces CV events, MACE events after an acute coronary syndrome or high risk. And we found that in the control setting, the HDL infusion was actually slightly pro-inflammatory, activating P20 formation, activating JNK. But in the TET2 clonal hematopoiesis, it really worked. So again, I would suggest that targeted therapy might make more sense than just giving it to everybody. And we used the drug holomycin, the deubiquitinase inhibitor, to show a benefit in TET2 clonal hematopoiesis. So again, this is lesion area, increased with the TET2 mutation, reversed by holomycin. Necrotic fibrous cap thickness, again, selectively TET2 deficiency holomycin, only in that setting, nothing in the controls. And the netosis was completely reversed by holomycin. And why should it be that TET2 and hypercholesterolemia interact so powerfully in this way? It's because we believe a convergent signaling occurs where the TET2 deficiency, TET2 promotes demethylation of genes, which tends to activate them. So that activates genes that are called DUSPs. They are dual-specific phosphatases that normally inactivate JNK signaling. And so in the deficiency state, the DUSP levels are reduced, and so phospho-JNK signaling is increased and converges with cholesterol loading effect to activate the NLRP3 inflammasome. So that's the new part related to TET2 deficiency. Okay, I've mentioned Mustafa and Trevor, Marit Westerterp, colleagues at Columbia. Peter has been educating me in the ways of inflammation. He's been working on it for more years than we can care to count. We've been working on it for about 5 years. Ross Levine at Memorial Sloan Kettering, Irene Papapetru, and I mentioned Z and Pradeep. Thank you very much.